This is Chapter 7, looking at patterns of inheritance or genetics. In this chapter, the book goes into a story about researchers and how they are unraveling the genetic secrets of domestic dogs. So we're going to have a couple examples of looking at genetics in relation to domestic dogs. I will also throw in a couple of other examples from humans or other organisms. So genetic traits are defined as any inherited characteristic of an organism that can be observed or detected. And genetic traits can be categorized in a couple of different ways. One way is by looking at if it's an invariant trait or a variant trait. An invariant trait, these are traits that are the same in all individuals of a species. If we look at domestic dogs, an example of an invariant trait would be domestic dogs have 4 legs. Another example would be domestic dogs have 2 ears, they have 2 eyes, 1 nose, they can bark, so that is an invariant trait as well. In addition to those invariant traits that are the same for all of the individuals, we also have variant traits. These are traits that differ among individuals of that species. So in our domestic dogs, we can have dogs that are large, medium, and small. So that trait differs. Another good example is fur color. We can have dogs with black fur, white fur, brown fur, red fur, spots, different combinations of these colors. So that's another variant trait. So we can have traits that are the same or traits that are different within our species. In addition, we can look at another type or categorization of genetic traits. Traits can be physical traits, so they're easily observed. So a lot of the traits that I just described in relation to the dogs are physical traits. So height, coat color, number of legs, we can actually see those and observe it. An example in humans is being able to roll your tongue or not being able to roll your tongue. In addition to these physical traits, we also have biochemical traits. These are hidden traits that are measurable. So these are things like insulin production. So some individuals have normal insulin production, whereas others have a lower insulin production or no insulin production at all. So that's an example of biochemical traits. Also our metabolism, some people have a fast metabolism, others have slower metabolism. A third type of genetic trait are behavioral traits. These are behaviors like being stubborn, like the little girl in the picture, and they have to have some genetic basis to them, so they can be passed down from parent to offspring. A lot of behavioral traits are also influenced by the environment in which the individual is exposed to or grows up in, and we'll look at that genetic versus environment and the influence on these different traits in a little bit. So all of these different traits, whether if they're invariant or variant, or if they're physical, biochemical, or behavioral, they are encoded for by a basic unit called a gene. So a gene is a basic unit of information that affects that genetic trait. And these genes, they consist of a stretch of DNA in our cells that code for a protein, and these are found in the chromosomes. So for example, our human body is made up of about 10 trillion cells. If we zoom in on one of those cells, one cell has 46 chromosomes in the nucleus. We zoom in even further into that chromosome and you have segments of DNA called a gene. And that gene will code for one specific trait. So these genes, we can have alternate versions of a gene, and these are called alleles. So different versions of a given gene are the alleles. So for example, we can have a gene that codes for tongue rolling, and we have one allele that allows that individual to roll their tongue. Another allele will code for not being able to roll the tongue. Or another example, if we look at the dogs, you can have fur color. You can have an allele that gives black fur, an allele that gives brown fur, an allele that gives yellow fur. So they're the same trait, there's just different versions of that trait, and those are the alleles. These alleles arise through mutation, so any change in that DNA sequence that makes up a gene is a mutation. And then that mutation will give us a different product or a different trait that is actually expressed. So here's an example of a mutation. We have our original DNA right here, or the correct copy. If we change one little segment of that DNA, that can change that gene. We can end up with a different allele, which could give us a different trait that is expressed. So the genes or the alleles that an individual has is called a genotype, or this is the genetic information that that individual carries. So an example from our book looking at domestic dogs, so genotype is coded for by little letters or big letters. So those are the alleles, and we'll get into what the big letters are and what the little letters are. But every genotype has 2 letters. So you can have a little b, which is what this blonde dog has for its genotype. You can have big B, or big B, little b. So you can have different allele combinations. These genotypes will then be expressed to give the individual's phenotype. That is the physical expression of that gene. So that is that physical trait, that biochemical trait, or behavioral trait, or how that individual looks or how it behaves or acts. So it's what you can observe in that individual. And these genotypes and phenotypes are very closely related because the genotype, or what genes you have in your cells, will determine your phenotype, or what you will physically express for that trait. So for example, here we have our 2 dogs. The blonde dog has a genotype little b, that will make the fur blonde. The black dog can either have the genotype big B, or big B, little b, and that will make a physical expression of black. So these 2 alleles in the genotype, in Mendelian genetics, which is the type of genetics that we're going to be looking at, this is very simple genetics, and we'll look more at that later. But those alleles, so one is called the dominant allele. So this is an allele that's expressed when there are 2 copies of it, or at least 1 copy of it. And it's expressed when there's 1 copy of it and 1 copy of the recessive. So usually the dominant allele is used or shown with a capital letter. So in our previous example, our dominant allele would be the big B. So we noticed that the dog that had a big B genotype, or a big B little b genotype, showed the dominant trait, which was black fur. That little b allele is called the recessive allele. So this is an allele that's expressed only when there are 2 identical copies of it, and it ends up showing that recessive phenotype. So the recessive genotype, or the 2 alleles, would be little b. In order to get that recessive phenotype, or that blonde fur color, you need to have 2 recessive alleles shown here. So these different genotypes that we've been looking at, we can further categorize them down into different categories. So we can have a homozygous genotype. These are the ones that consist of 2 copies that are the same. So if you have a big B is dominant. We have 2 big B's, so it's homozygous dominant. Or both of those alleles are the same and they're both dominant. You can also have homozygous recessive, which is little b. So they're both recessive, they're both the same. So these are 2 genotypes. The third genotype that we can have as a possibility is called heterozygous. So a heterozygous genotype is a big B, little b. And this consists of 1 dominant allele and then 1 recessive allele. So all of this information, looking at the dominant, recessive, we can have homozygous dominant, homozygous recessive, heterozygous. Those genotypes will give the phenotype when they're expressed. This goes back to work done by Gregor Mendel. So Gregor Mendel, he wasn't a monk. He's known as the father of modern genetics because he spent a lot of time performing genetic crosses on pea plants. So he took pea plants that had certain characteristics, he manually crossed them with each other to produce specific offspring. And he kept track of the parents, he kept track of the offspring, and he collected a lot of data. And based on this data, he came up with 2 laws that we're going to be looking at. His data showed that genetic crosses, we can use probability to figure out when something is going to be passed down from parent to offspring. Or these are called Punnett squares. Punnett square is a tool that we can use to predict the ratio of offspring expected by knowing the parent's genotypes. And in the lab, you're going to be doing a lot of Punnett squares, looking at just one trait to predict what type of offspring or the probability of different types of offspring and how they can be produced. So what Mendel found is when he took 2 pea plants, so if we look up here in this F1 generation, we have one individual pea plant here with the genotype big P, little p. We have a second individual here that also has the genotype big P, little p. What Mendel found was that during gamete formation, so during meiosis and production of those gametes, these 2 alleles will separate from each other and be put into separate gametes. So these are like the homologous chromosomes that are separated from each other during meiosis 1. These gametes are then put onto a Punnett square. So in this case, we have a 2 by 2 Punnett square. The female gametes, or the eggs, go on the side of the Punnett square, so she can either produce a big P or a little p. The male gametes, or the sperm cells, go across the top, so he can either produce a big P sperm cell or a little p sperm cell. Then what happens is you have random fertilization. So you pull one sperm cell and one egg cell into the box, you have fertilization. In this case, we end up with an individual that's big P, big P. We get a big P, little p right here if you pull in those 2 gametes. If you take this big P and this egg and you have fertilization, it's big P, little p. And then here you pull in a little p sperm cell, little p egg cell, and you get little p for the offspring. So these 4 combinations down here, these are the possible offspring. And these are the possible offspring that can be produced from these 2 parents up here that we started with. So using this idea and what Mendel found when he did these crosses and kind of worked out the numbers is we have Mendel's first law called the law of segregation. So this states that the 2 alleles of a gene, they're separated during meiosis and they end up in the different gametes, which are the egg or sperm cells. So for example, here, if we have a blue beetle that has the genotype big B, little b, right here, when this blue beetle produces gametes, that big B goes into one gamete, the little b goes into the second gamete. So those 2 alleles are separated or segregated from each other into different gametes. In the green beetle, we have little b, little b. So in this case, the little b goes in one cell, the other little b goes into the other cell or other gamete. So this law of segregation, this just states what happens to those 2 alleles during the gamete formation. Those gametes will then go through random fertilization to produce the different possible offspring. If you're looking at more than one trait, it can get very, very complicated. And this is where Mendel's second law comes into play, called the law of independent assortment. So this states that when gametes form, the 2 alleles of any given gene will segregate, and they will do this independently of any 2 alleles of other genes. So to kind of get a grasp on this concept, or this law of independent assortment, we can look at this genotype right here. So in this genotype, we have big R, big Y, little y. What this shows is that we have two different traits. So there's this trait big R, little r, that codes for one trait. The Y's code for a second trait. The big R and little r, those are just alternate versions of that gene or the alleles, but they still code for that same trait. So for example, if you're looking at fur color, so we can have fur color here, and then big R could be black, little r could be yellow, but it's still fur color. Another trait to go along with that, we could look at fur texture. So in this case, fur texture, big Y could be curly, little y could be straight. Still fur texture, just alternate versions of it. So here we have two different genes, one looking at fur color, the second looking at fur texture. So what this law of independent assortment states, it just states that fur color, those two alleles, will separate independently from the two alleles for fur texture. So this means we can end up with all possible combinations in the gametes. We can have a big R pulled with a big Y. We can have a big R pulled in with a little y. We can have little r, big Y. And the fourth possibility, little r, little y. So it just shows that those two dominant alleles, the big R, the big Y, are not linked with each other. They'll separate independently. The little r and the little y are also not linked, or they'll separate independently from each other as well. So what happens during this independent assortment is that we can get four possible combinations of gametes. So you can get four possible eggs. We can get four possible sperm cells. So our Punnett square will be a little bit bigger. It's going to be 4 by 4 or 16 possible offspring right here. And those sperm cells will randomly fertilize the egg cell. So you have 16 possible offspring that can be produced, but they will all have different combinations of the phenotypes. So in this case, we end up with some peas that are yellow, some that are green. We have some peas that are smooth and some that are wrinkled down here. So you get a lot of diversity by just looking at more than one trait. So you can imagine if you're looking at hundreds of different traits, it can get very, very complicated. And this can explain why we have so much diversity just within families. So if you think about if you have siblings, your siblings will be different from you, even though you have the same parents, due to all this mixing up that can occur during meiosis and then during that fertilization event. So these traits are referred to as Mendelian traits. So these are traits that are controlled by a single gene and they're unaffected by environmental conditions. So these are traits that are pretty straightforward. Mendel was very lucky that he picked pea plants because there's a lot of traits in pea plants that are controlled by a single gene, and the type of soil or the other environmental conditions do not change the outcome. So this goes into a theory called the chromosome theory of inheritance. And this is one theory that's really important for biology. We've seen another theory called the cell theory. So this is another big theory that explains a lot of things that we see in biology, and we can use it to form predictions. So this chromosome theory of inheritance, it identifies chromosomes as the factors that are shuffled and recombined during meiosis. These chromosomes are separated randomly into the sperm and egg cells during meiosis, which we just looked at in chapter 6. So remember, we talked a lot about genetic diversity and how those chromosomes are all shuffled up, different ways that they can be separated during meiosis 1 to produce all of that variation that we see. So there are a few variants to Mendelian genetics. So one is called incomplete dominance. So this is when neither allele is able to exert its full effect. So one allele is not necessarily dominant over the second allele. So it's a little kind of twist. So in this case, the heterozygous genotype is going to display an intermediate phenotype. So here we have an example coming from the book looking at dogs down here. So we can have a dog that has a genotype big I. That dog will grow up to be large. We can have a genotype big B, and that dog will grow up to be small. And these, the big I, and the big B genotype, those are both homozygous genotypes because both of those alleles or letters are the same. If we have a heterozygous, so if we have a big I, big B genotype, that dog's going to show an intermediate phenotype, so it'll grow up to be medium size. So this is kind of a blending of your two extremes in that heterozygous. Another little variation is called codominance. So codominance is when the effect of the two alleles is equal and it's visible in the phenotype of the heterozygous. So both of those alleles will be expressed at the same time. It's not like the incomplete dominance where both alleles are blended together. This is where those codominants, you can see both of them. So an example in dogs, gum color is a codominant trait. So a dog's gums can be pink. They have little p. So that is a homozygous genotype. They can have black gums or big P genotype. Or if they have the heterozygous, if they have big P, they will have pink gums with black spots. So here the pink and the black, they're not mixing together like the incomplete dominance we just saw. They're both showing up or they're both equally visible at the same time. So incomplete dominance and codominance are little variations in Mendelian genetics, but it can be even more complicated than that. So there's 3 complicated traits that cannot be predicted by Mendel's 2 laws. So we're going to look at pleiotropy, there's polygenic traits, epistasis, and in addition, there are environmental factors. So pleiotropy, this is when a single gene influences a number of different traits. So pleo means many, tropy means change. So you have one gene influencing a lot of different characteristics. So an example of here, the shape of a dog's head and the shape of its limbs. So we have 2 different traits or characteristics, the dog's head and the shape of its limbs. They're controlled by one single gene. So pleiotropy, one gene influences many different traits. In polygenic traits, this is kind of the opposite. We have a single trait that's controlled by multiple genes. So polygenic, or poly means many, genic refers to genes. So many genes control one trait. Examples of this are canine hip dysplasia. In humans, are hair color, eye color, skin color. So for example, skin color is controlled by 3 genes. So these 3 genes, they each have 2 alleles. So we actually have 6 alleles that control for skin color. So this explains why our skin color can show a very wide range from very, very pale to very, very dark in color. So multiple genes code for one characteristic in the polygenic. In epistasis is very complex. So this occurs when the phenotypic effect of a gene's alleles depends on the presence of alleles for another gene independently, or another independently inherited gene. So an example, in your book, they go through Labrador coat color, and I encourage you to kind of read through that and see an example of it. So looking at the different genotypes, it can explain or kind of add more variation. We can have black labs, brown labs, and yellow labs, depending on the genotype of 2 different genes and how they will interact with each other. In addition to those complex traits, looking at the pleiotropy, polygenic, and epistasis, we also have environmental effects that can influence the expression of those genes, which will influence the phenotype that's expressed. So there's a lot of external or internal conditions that can affect gene expression in genes. So one example that we find in cats and dogs in relation to fur color is ambient or body temperature. So we'll look at that one first. So here we have a Siamese cat, and in these cats they have darker fur at the extremities where their body temperature is lower. In the core of their body's a little bit warmer. So this temperature influences how that gene is expressed. So in warmer areas, that gene does not express that dark pigment. In the colder areas on the extremities, that pigment is expressed. So we have darker spots on the feet, the tail, the ears, and the nose. A lot of plants will have environmental effects. So for example, sunlight, CO2 levels can affect the color of plants or the color of flowers. Sunlight can affect the color of our skin. It can also change the color of our hair. So UV light, if we have exposure to that, eventually your skin will become darker if you don't get burnt first. And then that's the tanning part. So we have a lot of environmental effects that can change the phenotype. Our diet has a big influence on a lot of our traits, especially in relation to different diseases. Or different chemicals that we're exposed to can influence if genes are turned on or off, which will lead to different diseases as well. So most chronic diseases are very complex traits, and some of this can be controlled by different environmental conditions like physical activity. So here, for example, are different chronic diseases, so colon cancer, stroke, heart disease, diabetes, and the number of annual U.S. deaths from those different diseases. And you can see a lot of these diseases, about anywhere from 50% to like 20%, can be prevented by physical activity or looking at diet. So that environment can play a huge part in a lot of chronic diseases. So genetics, we have this genetic part. You can inherit the predisposition for this, but you can also look and see how the environment can influence these different traits as well.